So the animals that we're looking at are numerically the most abundant multicellular animal on the planet. Most of the organisms I look at are somewhere in the range of 1 millimeter to 5 millimeters in length. They're the primary food source for higher trophic levels, including all the commercial fish, potentially lobsters and right whales, and they feed on the primary production, algae and other smaller zooplankton. They can accumulate fat in these particular species such that they're 73 to 80% fat. If you took all of the copepods on the planet and you lump them up into a ball, they're equivalent to about 1 trillion human beings. And small changes then in the way that they function can have large implications for how the Earth works.
When we add dispersants to oil, we create these oil droplets, so we don't just have the dispersed oil in the water column, but also the oil droplets.
And actually, you'd only get those bubbles when you add the dispersant. Zooplankton wouldn't be eating the oil if there wasn't a dispersant layer put on it. Having an oil spill and then 4 days later thinking you've cleaned it up may not be correct, particularly if it's accumulating in these organisms. One of the ideas there is that these animals are exposed to a toxin. The toxin accumulates in the lipids. The animal goes down to the bottom of the ocean with that toxin in their lipids, and then they come back up to the surface and become a food source for the next season's young fish. And all of it requires sublethal levels. One in where animals are exposed to low enough levels that they survive, but high enough levels that it's got downstream implications. As you go up into the very top of the polar cap, it switches from the Calanus martialis and pinmarchicus to hyperborealis. And those organisms take 3 years to become breeding adults and reproduce. These long-term ones are difficult to do in a lab because keeping animals alive for that many years in a developmental cycle is a difficult one. But the deprivation part is huge. You'd be asking the question about whether if they're exposed and they go back down, do they get rid of their oils during their down periods? But all of these experiments center around levels that are not killing the animal. And those levels are much deeper down into the water column than I think are previously being thought about. Because they're sublethal levels. They're 1,000 times dilutions of the toxicities. Really, you're looking for minimizing the effect of either further treatment or is it better just to leave it alone?
What we hope is that the results of our study show whether there are seasons where adding dispersants to oil does not affect copepods. How do copepods react to oil? How is oil distributed through the water column? That this can be translated into a practical application and can go into the decision process when to apply dispersants. This information would be valuable for Coast Guard to decide when or when not to add dispersants.
We built these 2-meter tall towers and we introduced copepods into these towers, and then we put an oil spill at the top. And we let the hydrocarbons and some of the other toxic components of the oils diffuse down into the water column, and we look at the behavior of the animals in response to those oils. Which means they're still alive and they're still able to fundamentally change their relationship with the toxin that they're being exposed to. And one big question is always about whether they avoid it or not.
A next step would obviously be expanding this to larger mesocosm studies to include other species of copepods that are relevant for the Arctic food web.
Part of the direction that we'll be going, could be going, is to look at very, very low levels of oil products on the behavior of these animals and the long-term ramifications that it might have. And also to be looking at the possible accumulation of these toxins inside the animals themselves, these lipids. And at the end of the day, there is an oil spill. And somebody's got to make a decision about how you're going to treat it.
With this close contact with the ultimate consumer of science, it forces us to step out of our comfortable wheelhouse and think about how do we need to do our science so it helps